Good morning, everybody. My name is Nagachandrika Pallam and assistant professor, working as an assistant professor in pharmaceutics department, Sridatta Institute of Pharmacy. Okay, today's topic is introduction to the biopharmaceutics and pharmacokinetics. The subject of biopharmaceutics and pharmacokinetics for the class of B.Pharmacy third year, second sem. So, introduction to biopharmaceutics. What do you mean by this biopharmaceutics? First, we will know that what is the meaning of this exact meaning of this subject. Pharmaceutics is nothing but conversion of this dose, chemical constituent into a dosage form, that we call it as a not only that, with the safety and efficacy, the drug chemical constituent should be converted into a dosage form. So bio is nothing but the life, means that is indicate is the body. So how the drug is acting within the body, and how the drug is acting in the body, and not only that, how body is reacting to the drug, that we call it as a biopharmaceutics. And pharmacokinetic is nothing but the movement, pharmaco is nothing but the drug, how it is moving from administration to elimination site. So we are studying the dosage form after administering the drug, how it is acting to the body and how body is reacting, and not only that, how much after administration, how it is moving from administration side to elimination side, and how much drug is available while moving, how much of the drug is available, that we are studying in this biopharmaceutics. Okay, coming to these are the contents we are about to discuss: objectives for biopharmaceutics, definitions and pharmacokinetics, pharmacodynamics, and the bioavailability, different routes of administrations and references we are studying. Okay, so uh, coming to the objectives. So what is the main goal of this subject? Means to student will understand the concepts of ADME of the drug in the body. Not only that, to determine the various pharmacokinetic parameters from either plasma concentration or the urinary concentration of the data. That means while drawing the blood, that is nothing but the plasma, by drawing the plasma and the urinary samples, we are determining how the drug is moving from various parts of the body, that we are about to students will about to understand. Not only that, applying the various regulations related to developing the BA and BE study protocol. That means before the drug reaching into a market, the drug should approve from the regulatory authorities. So when the regulatory authority will approve, means after submitting this biopharmaceutical studies only, and if the drug satisfies the biopharmaceutical studies only, the regulatory authorities will approve. So these are the objective for studying this biopharmaceutics subject. Okay, coming to what is the actual meaning. So we know that the simple terminology we studied, but we have to study the technical way of definition. What is the technical way of definition? Means the study of various factors like physical, chemical, biological factors influencing the rate and amount of the drug that reaches the systemic circulation, and the use of this information to optimize the therapeutic efficacy of the drug product or the dosage form. You already know that the dosage form also called it as a drug product. So when after administering a drug, the drug should be reaches to the systemic circulation, that is nothing but the blood. Okay, so while reaching to the systemic circulation, various factors are influencing this drug to reach the systemic circulation. Some amount of the drug only reaching to the systemic circulation, otherwise some drug will be degraded, or full amount will be reaches to the systemic circulation. So not only that, with using this information, we are calculating, we are optimizing whether if it is degrading at any part, means we have to optimize, means increase our availability of the drug in the systemic circulation. So we are optimizing the drug why we are optimizing the drug? To show the therapeutic efficacy, means to treat the disease. That is only our main goal. With the safety and efficacy, only the drug should treat the disease. So that is our main goal. So this is you are unable to understand, I will show in next slide. So how it is actually, see, this is the dosage form. So after administering the drug, from administration side, if you are taking IV route, means it is directly reaching to the systemic circulation. But when you are taking to the oral administration, it should cross the various organs. So it should reaches to the through the mouth to esophagus to the stomach to it should reaches to the small intestine, through the small intestine through the portal vein it should reaches to the liver, through the liver the drug should reaches to the systemic circulation, means that it should reach the blood circulation. So availability of the drug means after administration of the drug, that we call it as a absorption. So from site of administration to systemic circulation, that we call it as a absorption of the drug. So after observing the drug, how it is so how much amount of the drug is observing and how much speed it is observing, that we have to calculate. So while reaching this systemic circulations, various factors, it should cross the various organs. That's why so many factors influencing the drug absorption. Not only that, the physicochemical factors, chemical factors, and biological factors and formulation factors. These are the factors influencing the drug. After here, CMA, after administering a drug, that drug is reaching to the systemic, reaching to the stomach. Through the stomach, it reaching to the small intestine. Small intestine through the portal vein, it reaches to the liver. So through the liver, it is reaching to the systemic circulation. Nothing but the blood. So after reaching the blood, it will distribute throughout the body on the site of action. It is distributing. So while distributing this drug, how much amount of the drug? So suppose you are taking 500 mg of the drug, that means 100% of the drug you are taking, and after reaching to the systemic circulation, only 250 mg or the 50% of the drug only available in the systemic circulation. That means in some organs the drug is degraded by the body reaction, otherwise the drug reactions, some drug is degrading. So we have to calculate how much amount, so how much you are taking and how much available within the systemic circulation, and not only that, how much speed it is reaching to the systemic circulation. That also very, very important. So if you are taking the drug, means it will delay for a longer period, means it will not show the therapeutic effect. So how much speed it is reaching to the systemic circulation, that we have to calculate. So how much amount and how much speed, both are very, very important. So here, while reaching to the systemic circulation, various factors are influencing. So here see physical factors, that physicochemical factors that we studied in industrial pharmacy 1. So biological factors, biological factors means after administering it, till that we are studied in industrial pharmacy 1. Now we are studying after administration how it is influencing the body, and body how it influenced by the drug, that we are studying in biopharmaceutics. And not only that, the way formulation factors also. So while compression factors, excipient factors, and various manufacturing factors also influencing the drug release, and some drug will not release fastly. So some, because of the compression, because of the binding agents, whatever you are using, the lubricants, because of the disintegrants. So these are the formulation factors also influencing the amount of the drug reaching to the systemic circulation and the rate of the systemic circulation. So while now till that most of the students they are studying till that only, Amma, but it is not ended here. So biopharmaceutics means by using this information, so of how it is reaching to the systemic circulation, how much amount it is reaching to the systemic circulation, how much rate it is reaching to the systemic circulation. By using this information, how we have to optimize our drug. If it is degrading means how we have to improve the drug. Okay, so not only that, so how much it is treating the disease. So if the 50% of the drug is available, how much it is treating the disease. Okay, and we have to design the dose regimen. Okay, how much dose we have to give to the patient, how much dose regimen we have to give, that we are deciding by these biopharmaceutical studies. Here dose is nothing but per day how many doses you are taking. Dose is nothing but how many tablets, means how much amount of the drug you are taking, that we call it as a dose. Means per dose regimen is nothing but per day how many doses, means 500 mg of the drug, how many times you are taking per day, that we call it as a dose regimen. So designing a dose regimen to design a dose regimen and the therapeutic effect, to know the therapeutic effect, we have to optimize the doses form. How we will optimize it means, if we know that these factors, if we study these factors, how it influencing the drug to reach the systemic circulation, how much amount it is reaching to the systemic circulation, how much speed it is reaching to the systemic circulation, we can easily optimize the drug. That is only the biopharmaceutics meaning. Okay, coming to the introduction to biopharmaceutics. So in detail I will explain, Amma. So after administering the drug, that drug is nothing but the drug product. That when we are taking IV route, it is directly 100% of the drug is reaching to the systemic circulation. Yes, this one is the systemic circulation is nothing but the plasma. So the drug is available in the plasma. So that we call it as a the drug administration site to systemic circulation, that we call it as a absorption. So that we call it as a absorption. Bioavailability is nothing but how much amount it is observing, how much speed it is observing, that we call it as a means rate and extent of the drug available within the systemic circulation, that we call it as a bioavailability. Okay, so after reaching to the systemic circulation, the drug should distribute through the extravascular fluids and to the site of action it should distribute. Okay, the site of action, it will after complete whenever it is reaching to the site targeted site, that when it is binding to that receptor of the targeted site, it will show the pharmacological action. After completion of this pharmacological action at the site of action site, again the after completion of the, it is inactive. So some the drug is not active anymore. So that when after completion of the action, it should return to the systemic circulation only. So see, after taking after administering a drug, that drug should reaches to the plasma systemic circulation to site of action only shows the pharmacology. When it binds to the receptor, then only it will show the pharmacological action. After completion of the action, it should return to the systemic circulation for elimination of the drug. Okay, so for the elimination of the drug, it should be metabolized. So that elimination will be take place. Extravascular after the drug should be reaches to the extravascular fluids and the site of action. Okay, these extravascular fluid and the site of action, we call it as a distribution. Okay, so this is the slide. Next one is, what is the biopharmaceutics mainly deals with the pharmacodynamics and pharmacokinetics. So pharmacodynamic. Pharmaco, already you know that pharmaco is nothing but the drug. Dynamic means action. We can say that this person is dynamic means how he is acting. So how is dynamically that person is dynamic means here we are studying about the drug. So how the drug is acting. Okay, so what a drug, after administration of the drug, how the what a drug does to the body, and means how the drug is acting on the body, that we call it as a mechanism of action. Okay, so here pharmaco is nothing but the drug, kinetic is nothing but the movement. So after administration site to after administration site to elimination site, how the body is reacting to the drug, that we call it as a pharmacokinetics. So pharmacodynamics mainly deals with the target site, how we have to it have to target the site, and how it should bind to the receptors, and how the protein bind to the proteins signaling pathways, and the mechanism, how it show the mechanism of action, how it is showing the mechanism of action, and physiological response, so how it is showing the response, and dose response we are studying in the pharmacodynamics. Pharmacokinetics, we are studying about the administration, absorption, distribution, metabolism, excretion, and urine data. By the urine data and plasma data, we are knowing all those things. And so how you are estimating means by plasma drug concentration. That is the main concept for studying the pharmacokinetics. Okay, coming to the ADME, I already told you the orally written. So this is the technical definition. So absorption is the movement of the drug from the site of administration to the blood circulation, that we call it as a absorption. D is nothing but the distribution. Here D is nothing but the distribution. Distribution means the process by which the drug diffuses is transferred from intravascular space to extravascular space. See, vascular is nothing but the blood. So through the blood to the body tissues, how it is distributing, how the drug is diffusing, and how it is transferring to the body tissues, that we call it as a distribution. Okay, metabolism. Metabolism is nothing but the chemical conversion and transformation of the drug into the compounds which are easier. Means to eliminate the drug from the body means the drug should metabolize. After metabolizing only, the drug should convert it into hydrophilic nature. So CMO, for absorption purpose, the drug should have the lipophilic nature. So to remove from the body, the drug should have hydrophilic nature. So to convert this lipophilic to hydrophilic only, the drug will eliminate from the body. So that means our biological system having some enzymes that will convert the when it is reaching to the liver, after showing the pharmacological action, it reaches to the liver. Liver, it binds the it will bind to the enzymes, and these enzymes, what will happen, these enzymes will convert into a hydrophilic nature. Through this hydrophilic nature, the drug is excreted through the body by the various cells. Here excretion is nothing but the elimination of a unchanged drug. It is more converting into a hydrophilic nature. Okay, so unchanged drug are the metabolite from the body via the renal, biliary, and pulmonary. These are the different organs. From these different organs, the drug is eliminated, excreted from the body. Okay, so bioavailability, I already discussed. Once again, I'm giving the technical definition. Bioavailability is the defined as the rate and extent of absorption of unchanged drug from its dosage form and become available in the site of action. See, this is the drug we are administering through the oral means CMM. If you are taking IV route means intravenous route means it is directly reaches to the systemic circulation. When it is reaching to the systemic circulation, it is directly distributing to the site of action. So in IV route, 100% of the whatever you are taking, the drug 100% is available in the systemic circulation, but it is not safe. all the time. So most of the pharmaceutical family dosage forms we are converting into a solid dosage form. This because this is the safest dosage form, and we can withdraw also. Okay, whenever we need to stop the dosage form, means stop the dosage form, we can withdraw the drug also. So that's why the oral administration is very safe. So because of this only, our pharmaceutical industry, 90% of the drugs convert, we are preparing in a solid dosage form. When you are taking a solid dosage form administration, what will happen? It will, after through the mouth, it reaches to the through the esophagus, it reaches to the stomach. Here the drug, here see the drug what you are taking, it will be disintegrated. Disintegrated means the drug is break down into a particles. That particles again it is dissoluted, disaggregated, and it is dissoluted within the stomach. The gastric medium, the drug is dissoluted. After dissolution, what will happen? The drug is throw out to the it will reaches to the small intestine. Through the small intestine, in the small intestine, you already know that is small villi like projections we are having, the membrane having the villi like projections. Through the villi like projections, the drug is transporting through this portal vein. See, this is the portal vein, it reaches to the liver. So after reaching to the liver, the drug is reaching the systemic circulation. So this is the CMO various organs. It should, while you are taking the oral administration, it should cross the various organs to reach the systemic circulation. So some of the drug is regraded. So if you are taking X drug 100 mg, so in the small intestine 70% of the drug only it is observing. So through the portal vein, it reaches to the 50. When it is reaches to the liver, it only 50% of the drug, means 100 grams means 50% of the drug only available. So that means this 50 mg is how it is treating the disease, that we have to calculate. So availability is nothing but bioavailability, nothing but the drug available. Here availability is nothing but the available, means the amount of the drug present, that we call it as a available. So where it is available in the body, here body is nothing but the systemic circulation. So in the systemic circulation, how much amount of the drug is available, that we call it as a bioavailability. Okay, so pharmacokinetic process. So what are the after absorption, what will happen? It after reaching to systemic circulation, it should be dispositioned. So these are mainly divided into two parts, absorption and distribution. Disposition, here after reaching to the systemic circulation, what will happen? It will disposition, means changing position. Position is dis, means changing. Okay, means position, the position is changing to various organs, means it is distributing to the various organs. So at the site of action it is reaching. So how it is disposing, means by distribution and elimination. Okay, so it is not staying in the blood, it is a liquid form, that's why it is moving from one part to another part. So it is distributed by the distribution to the site of action, and the elimination from the body. It is mainly two types. So the from in the elimination, it is mainly two types of metabolism and excretion. So after distribution of the drug to the site of action, it is showing the after completion of the pharmacological action, it should return to the so again it should return to the blood circulation. After blood circulation, this reaches to the liver. Here the drug is metabolizing. Why? Means it should convert into a hydrophilic nature. That's why the drug is metabolizing. It will convert into an inactive form, and the drug should be eliminated from the body. So that's why it is metabolizing. So after metabolizing, the drug is excreted through the kidneys. This is the pharmacokinetic process, means how the drug is moving. After absorption, the drug is disposition and how it is disposition means by distribution, by the elimination. So in the elimination, the metabolism, by the metabolizing the drug is moving, and by excretion it is completely removing from the body. So till the site of administration to elimination, the kinetics, how the drug is moving, that we call it as a pharmacokinetic process. Okay, so parameters. So how we will calculate this pharmacokinetic parameter? We already discussed pharmacokinetic and pharmacodynamics. So how we will estimate this pharmacokinetic and pharmacodynamics by these parameters only. How we will means by taking the plasma concentration versus the time. So that we call it as a, while drawing the plasma means that is nothing but the, while drawing the blood only, we can estimate how much of the drug available at various organs, that we can easily estimate. So how we are estimating means by this PTC profile. PTC profile is nothing but, I already discussed in previous my videos. So in that I clearly explained this drug plasma concentration, why we have to take on y-axis, why we have to take time on x-axis. I already in detail I explained. So it is dependent, that's why we are taking on the y-axis. Independent, we are taking on the x-axis. So after administering of the drug, see these, first you have to know about this MSC level and MEC level. So these are the MSC levels and MEC level. MSC level is that nothing but the maximum safe concentration, and MEC level is that nothing but the minimum effective concentration. Okay, so this is the after administration of the drug, how it is showing the action after completion of the absorption. The drug, how it is how it is removing from the body. So here see, after reaching the maximum concentration means 100% of the drug is observing. So after that the drug is eliminating. Here this stage with the absorption and elimination both are the same. So after complete absorption, it is immediately removing from the body. Okay, this we call it as a elimination phase, this we call it as a absorption phase. Okay, so how we have, what are the pharmacokinetic parameters? Here the parameters, first parameter is Cmax, Tmax and AUC. Means Cmax means maximum concentration. So how much? So whatever you are taking the drug, so 100 mg of the drug you are taking, so 100 mg complete absorption of the drug should be take place, means maximum concentration of the drug. Okay, we have to take, so that concentration we call it as a peak plasma concentration. Okay, and time, here see T max, how much time it is taken for the maximum constant to reach the maximum concentration, that we call it as a, how much time it is taken to reach a maximum concentration, that we call it as a T max. Okay, area under curve means that is nothing but the whole area, the absorption, whole area of the absorption, we call it as a area under curve. Okay, and pharmacodynamic parameters, minimum effective concentration. See, this is the minimum effective concentration. We also called it as a minimum inhibitory concentration. When we are calling as a minimum inhibitory concentration means when we are administering antibiotics, means antibiotics to show the minimum inhibition of the microorganism, how much concentration we need to inhibit the microorganisms, that concentration we call it as a minimum effective concentration. Okay, so minimum inhibitory concentration, minimum effective concentration, where for the drugs we are say that we are using this MEC, means minimum effective concentration. That means minimum drug to show the action, that we call it as a minimum effective concentration. So here see, onset of action also there. So minimum onset of action is nothing but whenever it is reaching the above the MEC level. MEC level means after administration. See, we are taking the drug and it is observing the drug within the body, but it for showing the action, it needs some amount of the drug. Okay, so for the single part only, it will not show the action. It will need some amount of the drug to show the action. So that minimum effective concept, that we call it as a minimum for effectiveness, we need some minimum concentration. That concentration, so to show the onset of action, it should need some minimum effective concentration. That means above the minimum effective concentration only we calculating the onset of action. Okay, so how much time it is taken for the onset of action, that we call it as a onset of time. Okay, and the intensity of the action, so to show the therapeutic effect, how much amount of the drug is needed, that we call it as a intensity of action. So how it is treating the disease, that we call it as a intensity of action. See, these above MEC level, we call it as a toxic level, means it will show the it will show the toxic effects. That means it will show the side effects. Okay, and minimum below the minimum effective concentration, that means it will not show any action. That is nothing but the sub therapeutic level. That means below the MSc level and means in between the MSc level and MEC level only we need for the therapeutic action. So to therapeutic action means to show the, to cure the disease, that we call it as a therapeutic range is nothing but to treat the disease, it should be in between the MEC level and MSc level and MEC level. So that range we call it as a therapeutic range. So see, these are the technical definition. Cmax is the nothing but the maximum concentration of the drug obtained after administration of the drug for the single dose of the drug, that we are calculating in the mg per ml or micrograms per ml or mcg per ml. So Tmax is nothing but time of peak plasma concentration. I already explained. Time required to achieve a peak concentration means maximum plasma concentration, that we call it as a Tmax. AUC nothing but the integrated means total area, how much area it is, how much from the small part of the drug, also how much drug is available within the plasma and how much time it is available, that we call it as a total integrated area under the plasma time profile, and the expressed in the total amount of the drug comes into the systemic circulation after administration. Okay, coming to the pharmacodynamic parameters, these are the minimum effective concentration. So minimum effective concentration, the receptors are the receptor site required to produce the therapeutic effect, that we call it as a minimum effective. So minimum concentration we need to show the action. So that we call it as a minimum effective concentration. Okay, so I already told you, when you are taking antibiotics, we call it as a minimum inhibitory concentration. And the maximum safe concentration is nothing but the concentration in the plasma above the, which is the adverse and unwanted effects are participated. That concentration we call it as a maximum safe concentration, means above this level it will show the toxic, and below this level is we call it as a maximum efficacy of the drug. Okay, so the concentration above the MEC level we call it as a toxic level. Onset of time, I already explained the time required to start producing the pharmacological action, that we call it as a onset of time. Okay, onset of action means the time begin to show the pharmacological action above the MEC level, that only we call it as a onset of action. Duration of action is nothing but the how much time, the period the drug is within the plasma, that we call it as a duration of action. Okay, and intensity of the action, that means minimum pharmacological response produced by the peak plasma concentration of the drug, therapeutic range of the drug concentration between MEC and MSC. This, that means how much, so it is showing the therapeutic action. So how much intensity, how much intensity of this action means how much it is curing the disease, that we call it as a intensity of action. So see, when you are taking different routes of administration, how it is available. See, different routes means parenteral, sublingual, inhalations, transdermal, rectal route, and the topical route. So when you are taking the intravenous route means 100% of the drug available within the systemic circulation. That means it is showing rapid onset of action. So intramuscular means 70 to 100%. That means large volume often feasible may be painful. Means because of this it is painful, that only it should reach the tissues muscular to systemic circulation. That means it should yeah, it should cross them. That's why it is degraded. Some amount of the drug is degraded within the tissues. So that's why 75% of the drug only, minimum 75 to 100% of the drug only available. Subcutaneous also same. And oral administration, CMA, it is main safe. Maybe it may be 5% to 100%. Means sometimes it won't show any action. Otherwise, 100% is available. Otherwise, 5% also. How much amount of the drug is available within the systemic circulation, it is depending upon the dose, how you are designing a dosage form, and how it is releasing the drug, and it is mainly depending upon the type of the drug. So it is more convenient, but it is we can withdraw the drug also. It is showing any side effects. If it is showing any side effects, we can withdraw the drug also, because that's why it is very safe route. And the rectal route, CMA, you can see when the pediatric patients, when the oral route it is not, if the patient is suffering from the fever means they will give to the rectal route. Means if through the rectal route the drug is 30 to 100% of the drug easily available. It can through the rectal route also we can easily remove the withdraw the drug. So this is a less first pass metabolism, means it is not crossing the liver. Okay, inhalation route 5 to 100%. So often very rapid onset. It is showing very rapid onset of action. When it is directly reaches to the systemic circulation, but it is showing the rapid onset of action. But we don't know how much amount of it is available. Here, see, here 100% is available in the IV route. That's why it is showing rapid onset of action. But here available because inhalation, while inhalating the drug, some amount of the drug will escape in the air also. So that's why while administering the drug, some amount of the drug can escape. So that's why but it is showing very rapid onset of action. Transdermal route, it is very slow. 80 to 100% of the drug is available, but the patient is in unconscious state. Otherwise, the patient is suffering with the ulcers or any if he's having difficulty with the other routes, we can give through the transdermal route. Okay, it will give them first, it will avoid the first pass metabolism, and it will give the slow absorption, but it will give the effective the therapeutic efficacy is more than the other systems. So here, see, this is a systematic diagram. This is the one after administering of the drug disintegrated the dosage form. From the dosage form, the drug is releasing. The drug is releasing means a breakdown of the tablet. We call it as a disintegration. Okay, breakdown of the drug, that we call it as a dosage. From the dosage form, we call it as a disintegration. After disintegration, the drug will be disaggregated, means for some fine particles it should be disaggregated. After that disintegration, the drug dissolve in the gastric medium, that we call it as a drug dissolution. After dissolution, it will take the absorption. After the absorption, it reaches to the systemic circulation, that we call it as absorption. Through the after completion of the absorption, it should be distributed through the various organs. After that distribution, it should be metabolized and excreted through the body. So here for the elimination site, it is after distribution the site of action. Here it is showing the pharmacological action, and after completion sorry, after completion of the pharmacological action, the drug should be eliminated again. Sorry, here I show the arrow marks refer. Okay, after it is excreted from the body. So here whenever it is accumulated within the body, it will show the toxic effect. So it is not binding to the site of action and it is accumulated within the body. That means it will show the toxic effects. After completion of the from this pharmacological effect, we can estimate how much the therapeutic effect, how much the toxic effect it is showing. Means how it is treating the disease, that we call it as a therapeutic effect. Toxic effect means how it is showing the adverse reactions or the side effects it is doing, that we call it as a toxic effects. So this we call it absorption and distribution and metabolism and excretion, we call it as a kinetic phase. Means administration site to elimination site, administration site to elimination site, the drug is moving from one part to another part. That means the drug is moving. That's why we call it as a pharmacokinetic. Biopharmaceutics is nothing but everything. The how much by using this pharmacological effect, we how much of the drug, how much of the disease we are curing, and that we can, by using this information, we can estimate or optimize the drug, that we call it as a biopharmaceutics. This phase, this therapeutic phase and toxic phase, we call it as a pharmacodynamic phase. Okay, so this phase is a clinical pharmacokinetics, means absorption after administration, how the body is clinically reacting and how it is showing the therapeutic effect and response also. After administration of the drug, how it is responding to the disease, that we call it as a the patient, how it is responding to the drug, and drug how it is responding to the patient, that we call it as a clinical pharmacokinetics. Okay, this is about the introduction of the pharmacokinetics. These are the references. So thank you for listening.